All right. Thank you so much for waiting, everyone. I'm super excited to show you Sample Manager. My name is Lance. I'm a product specialist here at LabKey. I help different labs and different organizations evaluate Sample Manager to see if it's the right fit for them. And then once we kind of do that evaluation process, I also help get labs fully onboarded and implemented so we can make sure that everyone's successful using the software. Before LabKey, I was actually a lab tech and an account manager for an infectious disease laboratory. And then before that, I also worked at an emergency vet and at a zoo. So I'm very familiar with different ways of collecting different information about samples. I've worked with different LIMS, Excel spreadsheets, paper records. So I'm very familiar with the pros and cons of each systems. And that makes me really excited about Sample Manager because I can see how we can help labs coming from these different systems and improve sample management in major ways. So today we'll briefly discuss what to consider when looking at a sample management system, who LabKey is as an organization, and we'll dive into a demo of Sample Manager. So from here, we'll probably discuss a little bit about all of these different features, but I want to kind of know what's important to you when evaluating a new system. So we have a poll that's going to launch. So we just want to know what features are you looking forward to in Sample Manager? It's multiple choice, so feel free to select as many as you want. We can look at sample tracking, freezer management, workflow, assay management, and electronic lab notebook. So I'll keep this up until we start the demo so we can kind of get more of a response. So when you want to choose the right system, there's a few things to kind of keep in mind. We want to know what different requirements you have for Sample Manager. That's knowing what your lab structure is. We want to make sure that we can support the number of users that you have. If you have any different organizational needs, like the volume of samples you're collecting, the types of samples. We also want to know how are you planning on capturing your data and importing it into the system? Do you want to do it through the software by importing spreadsheets like Excel, or do you want to do it through an API, which is communicating our software to other systems? We also want to think about the key features that you're looking for, and that's kind of related to the poll. Are you wanting something that's easy to use and flexible? Are there specific features that you need to have, like that freezer management or an ELN? Are you needing barcoding or tracking sample lineage so you know which ones are aliquots or derivatives, or maybe what animal that they came from? And then when you're evaluating a system, it's also important to consider the vendor. How does the vendor price their software? Is it a subscription based? Do you pay a one-time fee? Are there any hidden fees? Do they charge more for customer support or charge more for having different sample types? That's all super important to ask when you're looking at different vendors. But then ultimately, do you like working with that vendor? Can you see yourself growing with them or getting the support from them that you need? Do you click well with them? Having a better understanding of what's important to you and what your needs are can make sure that you're more successful in picking the right software. So kind of keep all of that in mind today as we look at Sample Manager. We'll kind of dive into who LabKey is as an organization. We're going to kind of give more of a history here. So LabKey started over 20 years ago as part of the Fred Hutch Cancer Research Center here in Seattle, Washington of the United States. And we're experts in scientific data management. A lot of us come from science backgrounds. So we're scientists building software for scientists. We've partnered with a wide range of clients from academia to pharma and biotech to government research organizations. So we've worked very closely with a bunch of different fields of sciences and ensured their success. With Sample Manager, we partner very closely with you. We help import your historical data, get your system configured the way that you want it, and we help train all the users that will be using that software. And then we also take your feedback very seriously. Our software is very dynamic and we're always growing. We actually have monthly releases with our software. So you're not going to get something that's outdated and won't be useful in a couple years. We're going to keep growing with you. And we want your feedback because we want to always grow in ways that you expect us to grow. So when looking at Sample Manager, there's three main pillars that we really focus our software on. That's data integrity, sample traceability, and system flexibility. So with data integrity, we're going to make sure that we're entering in our data correctly. We can set certain parameters for the data that we're entering. We have certain data types. So if you're entering a date, you can select a date. You can have drop-down options like text choices, just to make sure we're putting in everything as we expect it. We also have a very robust audit log for our samples, and that kind of leads into sample traceability. So we can track all the changes that have been made to your samples, but you can also track exactly where your samples are in storage, where they came from, and where they are in their life cycle. Then there's our system flexibility. We want to make sure that Sample Manager adapts to the way you work and you don't adapt the way you work to Sample Manager. So today we'll look at a system that's kind of set up for a more clinical diagnostics system, but just keep in mind, we can work with a variety of different sample types, such as dirt, rocks, blood, plants. We can also have as many different data fields as you need associated with your samples. So we're not going to limit what types of information that you're capturing. So today we're going to be looking at a bunch of different features with Sample Manager. And a lot of these features can be divided into two tiers. We have our starter tier, which is great if you're just getting started as a lab. This includes things such as freezer management, sample and lineage tracking, searching and reporting, and our full audit trail. And then there's for professional for labs that are a little bit more grounded and need more features. Professional includes everything in Sample Manager Starter, but it also has our assay data management, an electronic lab notebook, workflow management, data partitioning via projects, and programmatic tools like API so we can communicate with other systems. So depending on what your needs are, we can help make sure you pick the right tier. And if your needs change over time, it's super easy to upgrade from starter to professional. So we're always here to grow with you. And then we also have other softwares that go beyond these capabilities if you grow even more. So today, let's get started. If we're going to kind of go over into Sample Manager, but after that, we'll kind of come back here. We can get a personalized demo set up, answer more questions that you may have, and get started on the guided trial. But for right now, let's kind of go ahead and look at what our poll options are. So it looks like the majority of us are looking for sample tracking and freezer management. We also have some folks that are interested in our workflow, assay management, and an ELN. So we'll kind of cover all of those features today. And if you have any questions along the way, please feel free to drop a question in the chat or a QA. And I'll try to keep an eye on all the questions as I go along. So this is my system of Sample Manager. It's going to look very similar to what your system looks like. It'll just be a little bit different depending on the types of samples we have, the different sample statuses, and the different storage units. So on my dashboard, I can see a summary of all my different sample types and their statuses. Here I can see I have 10,000 DNA samples that are available, and I have 19,000 DNA samples that have been consumed. And that's true for all my different sample types. I can kind of get a look at them. I have a list of all my storage units. I can see the different types of samples that are inside each of these storage units. These are all color coordinated, so it's very easy for me to get a visual view of what's going on. I can also see the space that's available in these freezers. We try to make sure we tell you exactly where you can put a new sample in, so you're not having to go from freezer to freezer. Just try to find some empty space. We also have these statuses associated with these freezers, so you can indicate if a freezer's down, if it's defrosting, or if it's available, just to try to make it a little bit easier for you to keep track of what's going on in the lab. I have this list of jobs that have been assigned to me, so I know exactly what I need to be doing in the laboratory. I can also see what jobs are assigned throughout the entire lab. They all have due dates. And as you can see, we're not very good at doing our jobs here in the LabKey lab, so they're all overdue, and I need to do those as soon as I can. We also have a list of our recently opened notebooks, so I can easily get back to what I've been working on in the lab and kind of get back to my experiments. To navigate through Sample Manager, we have our drop-down dashboard here. This is going to pull up a list of how I'm organizing my data. So I have my different sources. These are things like animals, patients, studies, labs. It's going to be an umbrella of data that's going to be associated with your samples. And it also helps us know exactly where our samples came from. We have a list of our different sample types. So if we have like blood, urine, PBMC, plants, whatever it is that we need to track for our different sample types, we have it listed here. We have a list of all of our different assays. So this is where we can track our test results. All of our storage units, our workflows and notebooks are also here as well. And then we have our different projects here, which is how we partition data. So we can assign specific users to only see something from Project Alpha or Project Beta. But right now, let's go ahead and go into our blood sample type grid. This grid is kind of similar to Excel. So we have different rows with these different samples, and all of these columns have different sets of data that's important for the sample type. We can customize the data that's available for each of our samples, so we're capturing exactly what we need. Within each of these columns, I can filter out all different samples based off of the data that's here. So here I just searched for all my samples that have failed a QC state. If I had a larger list, I could then further narrow it down based off of any of these other columns. I can also see exactly where these samples are in storage if they're checked in. So let's just go ahead and find all my samples in storage. I can then see exactly where they're located and where inside of these boxes these samples are. From this grid, I can create derivatives and aliquots. These are samples that are taken from other samples. I can also add these samples to storage, add them to an assay run, and add them to like a pick list so I can reference them later on. I can also add samples to the system either by uploading them into the UI or through an Excel spreadsheet. Whenever you see one of these grids in Sample Manager, I can also export a file. So it's going to be a CSV, Excel, or TSV file, which is useful if you need to share this information with others outside of your organization. You might have also seen that we support printing labels. So if you need to print off barcode labels, we do partner with a third-party vendor that helps customize those labels and can print directly out of Sample Manager. For right now, let's go ahead and look at one of my specific samples. We're going to look at blood 4733. This sample just has a lot of data associated with, so I like to show it in my demos. When I click on this samples page, I can see exactly where it is in storage. I can see the amount I have available and its free saw count. I can see how many aliquots the sample has and how many of those are available. I can also see the metadata that's associated with the specific sample too. One thing that is really exciting for a lot of folks is our lineage view. So this shows how this sample is related to everything else in my lab. I can see that this blood sample was drawn from this specific mouse and that this specific mouse is from this specific study. I can also see that this blood sample has these 13 aliquots and then it has these derivatives here. So we went from like a blood sample to a plasma, PBMC, or DNA sample. And then I can then see those samples, aliquots, or derivatives as well. So this is kind of a great way for me to get a better understanding of how everything's related. And if I go to the animal level, I can also see all these samples that came off this animal. But you can imagine if you have an animal that's coming in multiple times, or maybe a patient, this might be a pretty big view and a bit daunting. So it's not that easy to see. We have the samples tab, so you can get more detailed information on all of these samples and how they're related to that specific mouse. So this is a great way for me to kind of get some more inside information. Going back to my blood sample, I can also see all my assays that's related to it. So I can see the different runs the sample's been a part of, the different test results we have. So this is kind of a great way for me to view things if they've changed over time. We can also view these assay tests on the source level. So if I want to see all my test results for a specific patient or a specific animal, I can have that information drawn up right here as well. One thing that's very unique about Sample Manager is how sample-centric we are, which means we really focus our data around the samples. A lot of other systems are freezer management. And the great thing about us being sample-centric is that we have a sample-centric audit log. So we can go to a specific sample and see everything that's happened to it from the moment we registered it in our system all the way to when we consume it. So here I can see that Hannah, one of our users, registered this sample back in 2023. I can see everything that's changed from it, what's kind of changed, who's done it, when they've done it. And then we can also have comments on these so we can see why these changes have been done. So this is a great way for me to go back in time and see what's happened in case we have an auditor that's come through or we have a quality event. And the great thing about this is that we also hold on to this view even after we consume these samples. So that way you can go back at any time and get a hold of these samples. A lot of other systems will delete this if you've discarded of a sample. For right now, let's go ahead and go back to the sample specific storage. And this is where we're going to kind of get more into the freezer management side of things. And if you have any questions along the way too, please feel free to drop them. I'm kind of keeping an eye out as I go along. So when I clicked on this sample specific storage location, it's going to bring me to this box map. This box map is going to highlight the exact row and column the specific sample's in, so it's very easy for me to get that visual representation. In this box, I can hover over any of the samples in here, and it's going to tell me the sample ID and the sample type. I can click around into these different samples and see more of that sample's metadata information, so I don't have to go back and forth between screens. So this is kind of a great way for me to manage my samples in these boxes. You might also notice that they're color coordinated. So this red dot is a blood sample and this blue dot is a DNA sample. We can change the colors of all of these different sample types so we can have a more quick visual of what's happening in our storage. If I kind of scroll down, I also have a box manifest of everything in the specific box. So if I need to ship these samples out, it's very easy for me to download this spreadsheet and send it to whoever may be receiving this box. From here, I can move my samples around. I can check them out, discard them. So let's go ahead and just check out blood 4733. I'm going to type in for my demo. This is going to be logged in my audit log. When I do that, I can see that this space is now highlighted pink. This means that we're reserving it, so we're not going to let someone else check in another spot because we're assuming when you're done experimenting on that sample, you're going to put it back in that location. If I want to free up the space, I'll just move it to like a workbench. Let's say now I'm done with my experiment. I want to check it back in. Sample Manager is going to ask you how much you used during your experiment. So let's just say we're using 0.5 ml. It's going to ask us if we want to increment our freeze thaw count. And I'm just going to type in my reason for doing this. So for my demo. And if we look right here behind the box, we can see we currently have 1 ml and a freeze thaw count of 16. Now, when I check in my sample, we automatically reduce the amount we have available of the sample, and we automatically increase our freeze thaw count. That way we have a real up-to-date timeline of what's going on with these samples, and we're keeping everything in real time. And we can have multiple users in the system at once. So if one user does it, it'll also update for everyone else. We can do this for one sample at a time, or you can select multiple samples and check them out, move them around, do whatever it is you need to do. So that way we make it very easy for you to either work in one single sample or in bulk. All of these boxes can be customizable to have as many rows and columns as you need. So if you have like an 8x4 box or rack, you can do that, or a 10x10. Whatever it is that you need, we can help support that. We can also customize these different freezers to have as many different shelves and racks as you may need, just to make sure that we're working with what your reality is. So here, when I'm looking at this freezer's kind of build, I can see on shelf 1, I have free space, rack 1 has this amount of free space. And we can see a lot of these boxes currently don't have any free space, but box 1 with these parentheses has 44 spots. So when you're checking samples in and out, super easy to find where you need to put these samples. Kind of going from our storage, let's kind of move over to our assays. So our assays is how we can capture experimental data. We can have data related to different runs, which is basically what's going to be all the information related to all of the samples that are currently being tested, which is usually something like a kit date or kit that's used, a run date, different operators, and a specific workflow task that we may be using. And then we can have result information, which is going to be what's going to be different between all the samples. Are they going to have different positive, negative results? Do they have certain genes that have been found? Speciation, whatever it is that we need to do, we can capture it here. We just basically enter in whatever our sample ID is, and then we can enter in the type of gene that we have. If, let's say, they all have the same gene, we can also do a bulk insert saying these 3 samples that I'm just now creating are all going to be gene 1. We can update it all at once, so you're not having to go one by one doing it. This is also a great place for you to use API to import your test results from something that's going to have more information that can export your data in a tabular format. Let's go ahead and move on to our workflows. And again, if you have any questions as I go through, please feel free to ask them. Our workflow is a way for us to track certain processes. It's kind of like a step-by-step SOP or standards of operations. So if I kind of go into my blood run workflow, I can see I have all these different tasks that I need to do where we receive the blood, check in tubes, we need to take 1 ml aliquot, then we need to run these different assays. These can be as detailed or as generalized as you need them to be. We can assign specific assays with them so we know exactly what kind of data we need to be importing. And we can also assign specific task owners. So if we have multiple technicians working on the same job at once, we can make sure that they're all going to be receiving those email notifications that they need to be doing something specific in the workflow process. And then they'll also get a notification in Sample Manager saying that they have a job done. We can assign due dates so we know exactly when they should be completed by. With each of these jobs, we can also assign certain data with them. So if we have like a shipment job, we can have tracking numbers, a specific courier company, and when we can expect these samples to arrive. We can attach specific files to these workflows. So it's another great way to have like an airway bill associated with them. We can assign specific samples to these workflows so we know exactly which samples we should be pulling. And this is also where we can look at our assays that's falling under the specific workflow. And this is where we can also import that data. These workflows can all have templates. So if you have specific processes that you're going to be repeating over and over again, this is a great way to do that and make sure everyone's following the same steps that they should be following. Now let's go ahead and move on over to our notebooks. So our ELNs are automatically going to be linked to our data in Sample Manager. And this is great because this is easy for us to reference specific samples, specific sources like a patient or an animal. So we can go back and see what we've done and we can check our data, make sure we're being accurate with what we're entering. These notebooks can have specific templates, so we make sure we're recording things the same way every single time, so you're not having to start from scratch formatting all of your experiments. When I look at my ELN, I can assign specific authors to it, so we can have more than one person collaborating on it. I can also, like I said, link specific sources or samples with it. And it works very similar to social media where I'm going to just use an @ sign and I can enter in a sample ID or a source ID. It'll be referenced in this notebook, and then this notebook will automatically be linked to that specific sample. These ELNs also support inline images, graphs, charts. So this is just a way for me to brag about my cat because I think he's really cute and I like to show him off. We have these tables that we can have within Sample Manager. So this is also a nice way to kind of format everything here. These notebooks can have multiple entries that are then going to be linked to this table of contents. So I can kind of jump around the notebook, which is nice if I have something that's very long that I need to be able to quickly get to. I can also kind of see where we're referencing these specific samples, click on it, get some more detailed information. And then I can also jump to where the sample is being referenced in the notebook. So this is kind of a great way for me to check everything. When I'm done writing everything, I can submit this notebook for review. The person that's assigned to review it will receive an email. They'll then enter in their electronic signature, verify that everything looks good. They can then submit it and say everything's great. We can download it as a PDF, share it as we need, or they can send it back to whoever the authors were to make some more changes if that's what's needed. So this is kind of a great way that kind of encompasses all of our data, getting our data in. But now how do we search for something? We have everything, but now I just need to find stuff. So there's a few ways we can search in Sample Manager. If you know the ID, you can search it right here. We can search through barcodes, so you can scan in your samples. It'll pull up a list that's very similar to that sample grid view. Or we have our sample finder. And sample finder is a great way to find samples based off of their lineage. So if I want to find all my samples that came from a male mouse, I can search for those samples and it's going to pull up a huge list of everything that's kind of meeting that criteria. I can then further narrow it down to find more samples of what I need. Let's say I want to find all my male mice that then are from a specific strain. I can further narrow down that list to find all those samples. If I'm going to be running these searches quite a bit for whatever it is, I need to pull a certain set of samples for an assay. I can save these and reference them later back on. Like I have a save search for all my expired samples. That way I can quickly pull a list of my samples I need to dispose of. But Sample Finder is a very powerful tool for you to use, and that's something that a lot of our current clients like to use. It helps them get back to what they're needing to do and saves them hours, if not days of time, trying to find specific samples. So that is kind of the gist of Sample Manager. It's a very high level overview. I'm more than happy to kind of answer any questions. But if you want to find more information, you can always go to our website as well. We have Sample Manager kind of listed out here, so you can read more about it. We do have our pricing listed here as well. We want to make sure we're very transparent, and we have our pricing published to make sure everyone gets the same exact fair deal. But if you have more questions, I'd be more than happy to schedule some time with you all to get a personalized demo so I can kind of judge it more towards your use case. If you're working more with animals, I can give you a more animal demo, or more humans, it could be more human demo. I can answer any questions you have. And then we also have these guided do a 30-day trial for free. We can help import a portion of your data so you can get a real use case of what Sample Manager might look like for your lab, and everyone can get familiar with it. That way you're not risking anything before buying the software. But for now, I'd love to open the floor up to any questions that you may have. So feel free to either drop them in the chat or in our question and answer section, and I'll start answering them. But after that, thank you so much for joining me. I really appreciate it, and I hope to hear from a lot of you soon. Right. So one of the questions we get quite often is, how long does it take to get implemented in a new system? Which is always a great question. A lot of LIMS take months, if not years, getting started. With Sample Manager, though, with our full customer support, we typically say that it takes 4 to 6 weeks to get fully onboarded. And that includes getting your system configured to all the different sample types you have, the different types of data you need to capture. It includes importing your historical data, and it includes training all of your users. Of course, that timing can change depending on how messy your data is and how often you're able to meet. But 4 to 6, 6 to 8 weeks is typically normal. And our customer service is included with that, so we don't charge any extra for it. We help onboard everyone. If you have any questions or any issues that come up after the implementation process, we're still here to help, and we're more than happy to meet with you for as much as you need to. A lot of folks also ask, do you limit the number of samples types that you have, or limit the number of samples? We don't limit that. We want to make sure that you're able to capture everything that you need. So we can really make sure that we have the different types of data you have and the different types of samples that you need to work with. All right. Some other questions that we may sometimes get is, how easy is it to learn Sample Manager? What resources do you provide? If you kind of do a demo with me, I can definitely search. We do have some great documentation for you to look at. We also have different videos that you can watch to learn the process. But one of the main focuses we have with Sample Manager is making sure that it is user-friendly and very easy to learn. So it's not that complicated of a system to kind of get everyone used to. And then some other questions we get is, do we have to use this sample ID when we're doing it? So we can customize what naming patterns you have for your different sample types. So that way, if you don't want to use blood and a random number on it, you want to draw something from like the collection date and whatnot, we can do that. And great, another question. We generate data with Excel. Is it easy to upload with Excel data? Yes, so it is super easy to upload with Excel data. So I'll kind of show you how we do that. So the data that we're going to be importing from Excel should kind of follow the same sort of template that we have set up with Sample Manager, and that's something we can help as well. We also provide a template. So if I kind of download that, it's going to download an Excel spreadsheet for me to follow. Once I have all my information in there, if it's going to be like my source, my assays, or my samples, I can import through an Excel spreadsheet. I basically click, drag, and drop, and then it'll upload that data for me. But that is a fantastic question. Thank you. Are there any other questions? And if you think of any other questions after the webinar, we'll be sure to follow up, and you're more than welcome to email me. I'm always happy to answer, always happy to meet, make sure that we can help you out with whatever it is that you need and make sure we're a good fit.